welcome indeed to this afternoon's webinar. I am delighted to introduce you to Dr. Michael Morten from Imperial College London. I see we've got a few of your colleagues here in the webinar as well. And yeah, we're really looking forward to kind of seeing the innovative things that you've been doing with Amytracker. So thank you very much, Mike. Over to you.
Thank you very much, and hello to everyone. Just trying. So yeah, today I just want to thank the organisers, obviously, for giving me the opportunity to present. It's really exciting to be able to talk to you about my work characterising protein aggregates, and it's nice on a level because we've worked with Ebba Biotech and used their Amytracker dyes to develop the fluorescence microscopy that we're going to be just talking about today, and going to describe how this has allowed us to study neurodegenerative diseases. And sort of as a bit more of a rounded introduction, I guess. I'm Mike. I'm a postdoc in UE's lab at Imperial College London, and obviously it's always sunny in London, but I only ever get to enjoy it when I'm not doing microscopy work in a dark room. And here I just put up an image of our turf microscope where we used to study aggregate species that are associated with different types of dementia. And I know that in other episodes of this webinar series, other people have sort of introduced this aggregation pathway before. So here we're just going to think about it in terms of a relatively simple process where misfolded proteins come together, they coalesce, they go through these structural rearrangements, and they form these beautiful, very stable, strong structures with this beta sheet stacking going right the way across the length of these aggregate structures. And these are a hallmark of many different types of dementia, including Alzheimer's disease, Parkinson's disease, frontotemporal dementia, and dementia of Lewy bodies, to name a few. But they're not the only hallmark. And the important thing to note here is that none of these hallmarks exist in isolation, and they're all sort of connected somehow. And just to sort of give you a brief journey through some of them, we can see how decline in proteostasis is linked to accumulation of protein aggregates, and these in turn are able to disrupt the mitochondrial function. They are sort of responsible for triggering inflammation pathways. They, again, disrupt synaptic function, and all this sort of overwhelms the cells and causes this loss of neurons in the brain during the progression of these diseases. And it's really important for us to be able to, again, look at how these are connected, but also in a way that it just goes to show how complex these diseases are. And the way that we think about classifying these different diseases is through the proteins that are more commonly found in the deposits in the brain tissue. And here I've just thrown up three different examples. We've got alpha-synuclein for the synucleinopathies, the tauopathies, and the proteinopathies that are associated with the aggregation of amyloid beta. But the closer we look at these diseases, the simplicity of this classification breaks down. And the famous example, I guess, is Alzheimer's disease, where we know that it's linked to the aggregation of amyloid beta in the extracellular plaques we see, but is also linked to aggregates of hyperphosphorylated tau in the neurofibrillary tangles. Now, you would thought this puts this squarely in the overlap between amyloid beta and tau in this particular Venn diagram. But once we start to take into account the fact that patients who score positive on seed amplification assays for alpha-synuclein unfortunately experience an accelerated disease progression. So that's why we've sort of nudged that Alzheimer's disease position to sort of in the middle of this Venn diagram. And we can spend forever sort of debating the sort of relative positions of these types of diseases. But really the take-home message of this slide is that classifying these diseases is very complicated, and what we need is very nuanced and sensitive ways of detecting them, so that that we can detect them early, because the earlier that we detect them, the better chances of any sort of potential therapy having on modifying the disease. But also we need to detect them and classify them accurately. So the canonical way that people have done this historically, including Alzheimer himself, was using microscopy. And here I've just put up an example from the Vandenberg lab, where they've used immunohistochemistry in this particular figure to show that the different antibodies that are targeting different epitopes of alpha-synuclein are exhibiting themselves in all these different kinds of deposits throughout the brain. And this is in Parkinson's disease itself. And if we zoom in and just look at the molecular arrangements of these types of deposits, here I'm going to show you the different molecular folds that we see in the tauopathies. We can see there's this rich sort of variety between the different diseases. Some folds are similar across diseases and some are different. And this is again mirrored in the synucleinopathies, where we see Parkinson's disease and dementia Lewy bodies having a near identical molecular folds. But when we compare this to multiple system atrophy, we can see that this breaks down and we see a very different fold, even though the protein within these aggregates is the same one. So it's not particularly a very simple one protein, one disease or one fold model, but there's a rich variety of structures that we're able to see, both on the microscopic scale and the atomic scale. But there's a missing sort of regime here in the middle, which is the sort of the nanoscale. And this is very important because there is a growing amount of evidence showing that these are the aggregates that can be measured in terms of nanometers are the ones that actually have quite a significant role in the pathology of these diseases. This is a study from the Kleneman lab, who illustrates this quite nicely. And what they've been able to do is fractionate the different aggregates using a sucrose concentration, and have measured the size of the aggregates using TEM, and shown that it is actually the smaller fractions that produce the bigger responses in terms of TNF-alpha release and are able to disrupt liposomes to a greater extent compared to the larger fibrils. And this is wonderful, but what we wanted to do really is use a technique that was compatible with live cell imaging, which is why we chose fluorescence microscopy. And here our initial experiments that we started off doing were with recombinant proteins. And this is where we took the proteins and we aggregated them ourselves in the lab. For example, with alpha-synuclein, we used shaking conditions at 37 degrees over a number of days to produce the aggregates that we're interested in. And we immobilized them on our cover slip, and we labeled them either Alexa Fluor 488 or 647 probes, or use the aggregate-specific dyes such as THT. And then the really exciting thing at the start of this project was that we were able to find there was this new generations of THT-like dyes where they were aggregate specific. And this included the Amytracker series and also the proteostat dyes as well. So what we did was we took our recombinant aggregates and we imaged them using a THT and a combination with either the Amytracker or the proteostat, just as a sense check to make sure that we were imaging what we thought we were imaging. And we can see that on the right-hand side here, we've got aggregates that are extremely long and quite large. And I think this is one of the largest that we've imaged in the lab, about 30 micrometers. And we can take this and we go all the way down to these aggregates, which exist in sizes that are close to the diffraction limit of light. And it is at this point that we start to lose some of our resolution, our resolving power, and we can't really make out the structural features in any great detail anymore. So in order to push through this diffraction barrier, we use single molecule localization microscopy. And I'm not going to spend too much time on this, but basically it relies on the fact that we need to be able to detect single fluorophores. And once we've pushed, and we do this by pushing the fluorophores into a bright state and a dark state, and once we get an image of a single fluorophore, which you see here on the bottom on the left, we can fit it to point spec functions and then calculate its localization to a greater precision. You can see this happening in the simulation on the right, where if we record all the emission from the fluorophores all in one go, it's kind of blurry and we can't really see what's going on. We start blinking and we start noting down the localizations, and we get a much more clearer idea of where we are and what we're doing. So in practice, the precision that we can calculate increases with the number of photons we collect from each fluorophore. So what we did was we detected thousands of fluorophores. And this sounds impressive, but because they're very small, it didn't take that long, to be honest. And then we compared the Alexa Fluor 647 and Alexa Fluor 488 dyes, which are the sort of the gold standard dyes that we use in single molecule localization microscopy. And we use this to set a benchmark to test the other aggregate specific dyes that we're interested in. And we can see that compared to the THT, which is sort of the traditional dye that we use as an aggregate specific dye, we see that the Amytracker and the Proteus dye actually outperform them and are much more similar precision to the Elexaflor dyes rather than the THT. So this was really encouraging. And to show you what the images actually look like, you can see here on the bottom half of this image, we can see that aggregates are kind of blurry. We know they're sort of there and that they're fibrillar, but we're kind of all the same. But once we do the single molecule localization, we can see a much more sharper detail of the fibrillar sort of structures. And this gets more interesting when we zoom on the smaller aggregates that are below the diffraction limit of light. And just to sort of convince you here that what this allows us to do, even though we're doing very simple sort of shape analysis and we're just measuring the size of these aggregates, we can get a lot better idea, especially when it comes to the very small aggregates that are shown here down in this single molecule sample. And that's great, but what we wanted to do was look at the biology of these samples. So we added aggregates to hex cells that were expressing proteasomes labeled with GFP. And the diffuse nature of the proteasomes meant that we could use that signal as a sort of like a pseudo cell mask. And this meant that we could see which aggregates were able to penetrate through the cellular membranes. So for example, here we can see these rather large aggregates sitting on the top of the cell periphery compared to these like sort of orangey yellowy ones, which have been able to penetrate inside the cell and sit like pretty obviously inside the cell volume. And from these images, we were able to measure the particular sizes of these aggregates. And here we can see the relative frequency of these sizes. And we can see that the aggregates that were able to penetrate the cell membrane at 4 hours showed a lot larger proportion of very small aggregates compared to the ones that are internalized after 12. And we interpret this crossover here between the 4 and the 12-hour lines at 450 nanometers as a threshold of aggregate size, which allows these aggregates to more easily penetrate into the cell. So we decided to take this a step further, and we wanted to see if this had any sort of disease relevance. So we took different samples of aggregates that we, again, this is all recombinant aggregates that we've aggregated together in the lab. And we've looked at the different size distribution using this single molecule localization techniques. And we can see that if we stop the aggregation reaction after 12 hours and measure the different sizes, we see a very different size distribution than if we let the aggregation curve carry on to 36 hours, for example. So here we have a much more larger proportion of samples under this 450 nanometer threshold. And then we took a second sample that we'd let to go to the 36-hour endpoint, and then we sonicated it again to increase the number of these very small aggregates. And you can see here when we do an LDH assay, which measures the cytotoxicity that is that each of these samples were able to introduce, we see here we get this very linear relationship between the fraction. If we normalize the concentration of the proteins in terms of which aggregates are smaller than 450 nanometers, we get this very linear relationship between the LDH response. So this was wonderful. We were very excited about this, and we wanted to test whether or not this had any particular relevance to real aggregates that we get from brain tissue from donors who've been diagnosed with particular neurodegenerative diseases. So here we have a very similar experiment done with aggregates that we've extracted from brain tissue of donors who are diagnosed with Parkinson's disease. And here I just throw up a very simple methodology where we take the brain tissue, and it's actually a very gentle extraction procedure where we just soak the brain tissue with artificial CSF. We spin it down to remove all the debris, and then we use ultracentrifugation again to spin down. And it's this supernated from the ultracentrifugation is that what we're interested in this particular experiment. And we can show, again, if we look at the cytotoxicity response at just the sort of total aggregate concentration, we get very different profiles. But if we exclude all those aggregates that are larger than the 450 nanometers, and we normalize our aggregate concentration to just represent those aggregates that we think easily penetrate the cell membrane, what happens is these profiles converge, and then we are able to see what could be a disease-specific profile. And again, we tested that again with Parkinson's disease patients and dementia with Lewy body disease patients, and again with donors who haven't been diagnosed with dementia. And we saw that what our suspicions were correct, and we saw that the Parkinson's and DLB and control and these donors who weren't diagnosed with dementia all had very different cytotoxicity trends when we normalized the aggregates to these aggregates smaller than 450 nanometers. And this was really encouraging, and we thought, but we didn't really understand why the differences that we were observing, but we thought that conformation may have something to do with it. So, and again, we're not the only lab who's interested in this, and we can see that other groups are using fluorescent microscopy to sort of also explore this research question. And this is a very impressive microscope setup where they've managed to get the resolution down to sort of like under a nanometer. But we were sort of struggling to figure out how to robustly and comprehensively quantify the aggregate morphologies. And during the sort of the lifetime of this project, we've actually been acquiring lots and lots of aggregate samples, and we have over 1 million images of aggregates from different types of dementia. So not only have we got PD and DLB, but also we have the aggregates that were extracted from brain tissue for people who have been diagnosed with Alzheimer's disease and FTD, as well as their age-matched controls. And because we have this vast library of aggregate samples, we thought that machine learning would actually be a very good tool to use in terms of quantifying the aggregate morphology. So here, and this is the work done by Haley and Eva in the lab, who've been able to really help out with the dry lab aspect of this project. And we sort of identified that autoencoders were actually really good tools to use to solve this particular problem. And the way this works is that it has 2 different neural networks either side of a latent space. And what happens is that we take each particular aggregate image, the encoder, this first neural network, sort of condenses all that information down into as we interpret them as morphological features. But all the information that it needs to reconstruct this image is done by is held here in the latent space, and then the decoder recreates the image that it was originally put in the input of the encoder. And then as we go through all these different images, these particular models learn better and better about how to reconstruct them. And here I'm just going to show you one example. It's for a very small aggregate, but it's like really nicely displays exactly how this works. But the really useful thing is that we have now a quantification of every single aggregate that we push through this model. And we can use UMAP projections to sort of reduce the dimensionality of this information and project it on 2 axes. And then we can subject this to k-means clustering, which groups together different pools of aggregates that all share similar morphological features. And then we can look at this subpopulations of each cluster and use this to try and predict whether or not a particular sample would have Alzheimer's disease compared to a donor who was not diagnosed with Alzheimer's disease, for example. And this is done in a one-to-one model. So this is like a reasonably simple model, but what we're showing here is that the accuracy of these models increases with the number of clusters that we're using. And we can see that by the time we reached about 90 different clusters to cluster all these millions of aggregates together, we're actually achieving an accuracy of over 95% across all 4 models, where we're trying to predict whether or not they have Parkinson's disease versus age-matched controls. And this is done using logistic regression analysis combined with recursive feature elimination. And this is wonderful, and it's really encouraging that we can use morphology in this way, but it doesn't really tell us what morphology features we're looking at. So we used a second analysis, a Dirichlet analysis, to pull apart the different clusters and just find out which ones are significant. And the Dirichlet analysis is really useful in this context because we're looking at the different fractions of aggregates within each cluster. So if we observe an increase of aggregates in a particular cluster, we would expect a corresponding decrease amongst other clusters to sort of cancel that out. And what Dirichlet analysis do is it sort of robustly identifies the statistically significant increase and decreases within each cluster. And here, what this has done is highlight several different clusters for each disease. And here I'm just showing you one cluster per disease, which have been identified by this Dirichlet analysis to be associated with that specific disease. But still, we don't really understand why the neural networks have identified these particular morphology features as important. So what we did was, again, we put our thinking caps on and figured out and scoured the internet and tried to find some methods in how we could interpret this. And we came upon this package called IMEA. And what this holds is a Python package which defines, I think it's over 50 different morphology features or 2D shapes. And some are more intuitive than others. Some are very simple, like the area, perimeter. But things like the geodetic length is like, it's not quite, at least to me, it's not quite intuitive. And then on top of that, we added on 5 different shape features that we've known to be used in the past that describe aggregate morphology. And what this allows us to do is if we measure the different aggregate clusters, we can see that for cluster 3, for example, which is associated with FTD, we can see that it has a very high scores when it comes to the number of branches in its skeleton. So, but then it has very low scores in terms of its thickness, essentially. So what we think is happening is that this particular cluster, which is associated with FTD, involves these sort of like long-ish branches, which have lots of little shorter branches coming off from that main branch. So that's really encouraging, which basically means that we've got this this way of quantifying morphology and figuring out what the different morphological features are. But it's not the only interesting feature of these aggregates. And composition of aggregates is also very important. So we've started to use immunofluorescence to tag the different aggregates, just to confirm that they are actually the aggregates that we're interested in, and what we're seeing is real. So we have this sort of joint labeling using Amytracker, as well as the different antibodies against. Here we're showing amyloid beta in red, and tau in green, and alpha-synuclein in blue. And we can correlate the frequency of these labeled aggregates with the neuropathological scores that we get from the brain back when we've that we get all these brain tissues from. And this is just a very quick, and it's a working process. And bear in mind that it's a very gentle extraction. So we're comparing, and then we want to move on in future work to sort of compare different extraction techniques to see if we get any different correlations in these brain samples. But it's a really exciting project. And I just want to finish off by giving you a very quick case study in terms of an experiment where we've taken brain tissue that was from a donor who'd been diagnosed with Parkinson's disease, again, extracted aggregates from it, and then added proteasomes to these aggregate samples. And we've used our single molecule localization to image both the reactants and the products of this reaction. And then we've put it through a very similar pipeline where we've used classical shape analysis to measure the features. We've used UMAP, dimensionally reduction, and K-means clustering to identify different clusters. And we can see that the brain soak, we've termed this the brain soaking aggregates plus proteasome samples, are pretty uniformly spread across this UMAP projection. Here we've only got a couple of thousands of aggregates compared to the millions that we had in the other libraries. But still, we are able to identify 10 different clusters within this aggregate population. And the important or interesting things that we've noticed is that when we add our proteasomes and they are able to degrade these aggregates, is that these proteasome samples have a very, or at least a pronounced, significant change in the number of smaller aggregates within each cluster. And then, interestingly enough, the reactant products left over from these proteasomes, as they've been able to fragment and degrade these these aggregate samples. The ones that are left behind, the ones that are potentially more resistant to degradation, have a higher eccentricity score when we look at the smaller ones compared to the reactant products, compared to the reactants at the start of the reaction. And this is interesting because it kind of agrees with similar studies or similar characterizations done. Again, this is the Penniman lab, where they've looked at tau aggregates. And again, they've scored these on the eccentricity too, and they see that there's similar higher degrees of higher eccentricity scores, and these were relevant aggregates. So it's really interesting to note that the proteasome degradation resistant aggregates here in our lab are sharing morphological features with the aggregates that are popping up in the dementia related sorry, that have been extracted from donors that have been diagnosed with dementia. So I think that's just coming up to about my half hour, but like, I hope you've enjoyed this seminar, and just the three sort of big takeaway messages that I hope everyone can sort of appreciate would be that single molecule imaging using these next generation of dyes is a really good way of characterizing these aggregates at a higher resolution that is capable of using traditional microscopy. We can understand the differences in morphology using very sort of classical and traditional methods, and this can give us some ideas about the disease-relevant properties of these aggregates, but also that if we embrace sort of like higher and more complex and deep learning strategies in terms of quantifying the aggregate morphology, this possibly opens up opportunities to really delve into whether or not into what's going on biologically, and whether or not disease-modifying treatments will be working, for example. But it's just a really exciting opportunity here, and I hope you've all sort of enjoyed this seminar, and I just want to thank you for taking your time out today, and obviously thank you and all the members of the eLab as well, both past and present, especially Haley and Eva. We've had a lot of support from Alan Murphy, who's a PhD from the Nathan Skeen group here at Imperial, and again, special thanks to all the donors and their families. Obviously, without this, would not be possible if they didn't decide to donate their brains for this work. So yeah, thank you very much, and I'll be happy to take any questions.